Now let's shift to the next family called tyrosine kinase inhibitors. This is a brand new family. This new family is so much classy with its expensive drugs. I know you'll like it. You'll see that people who made this family are very smart. But before we dive into details, let me remind you of this photo which we discussed in the previous video, the introduction. I told you cancer cells release something called growth factor. These growth factors work on a family of receptors called tyrosine kinase receptors. When tyrosine kinase works, it activates a chain of proteins. We call this chain signaling pathway. And it ends with activation of a protein within the nucleus which activates the DNA. Then the cell divides endlessly due to activation of its proliferation machinery. This is what we discussed the last time. This is ordinary. This is what happens naturally. It is natural for cells to release growth factors and activate other cells. This is natural. We all have insulin growth factors and so on. We all have these growth factors. So where is the problem? The problem arises when this receptor, this one, gives us attitude. It is activated by these factors and should stop when they stop. However, this time, once it's activated, it stays active even when these growth factors disappear. This receptor is determined to keep working and activating all of this chain forever. This is where the problem comes from. But why did this happen? This is genetic. There is a genetic error or mutation as we discussed before. When I told you that sometimes a mutation occurs in some cells, and when this happens, the gene changes into oncogene, resulting in cancer. This is a quick reminder for you so you can understand this family called tyrosine kinase inhibitors. This idea is based on the concept we discussed on the previous slide. What are this new family called tyrosine kinase inhibitors? They are drugs ending with NIB, N-I-B, such as Imatinibe, Erlotinib, or anything NIB. Any drug in the world ending with NIB should draw your attention because it's one of this new family called tyrosine kinase inhibitors. I want to focus on this one, Imatinib, because it is the most common one and it is the one you will see in exams. So please keep focused on this one. On the personal level, when examiners ask me to put a question or two about cancer chemotherapy for the students, I am telling you, I always choose this one. I like to ask in this one Imatinib in order to understand its mechanism of action and what ideas brought it to life. I want to show you this drawing. Keep focused on this part because it enables you to understand this new drug Imatinib, which we use for treating so many types of blood cancer or leukemias. We have chromosomes in our body. Among them is this one, chromosome number 9, and this one, chromosome number 22. These two chromosomes, sometimes when our body cells divide, these two chromosomes are known to have some issues. Before I tell you about the origin of this problem, firstly I want to clarify something. Chromosome number 22 carries a gene called BCR. Chromosome number 9 carries a gene called ABL. I don't care about their function, I don't know, and I don't need you to know it. These two genes can have any function. In some people, during cell division, an error may occur during DNA duplication at S phase. This leads to separation of this part from chromosome number 22 and separation of this part from chromosome number 9. This detached part from chromosome number 9 attaches here to chromosome number 22 and this part attaches here. This replacement is termed translocation. Scientists know exactly what it is. The first time they discovered it was in year 1960. The two American scientists who discovered this were from a place called Philadelphia. So they called this new chromosome number 22, which took part from chromosome number 9. This new resulting chromosome is called Philadelphia chromosome, after the city where it was firstly discovered. They also discovered something they weren't aware of before. They found out that these two genes, BCR and ABL, remember that this gene was here and this gene was there. This part detached, then attached to this chromosome, forming Philadelphia chromosome, resulting in two genes sticking to each other. This red one and this yellow one, forming a new gene. What did they call it? They called it BCR-ABL. This new gene causes a very big problem. Where does this problem come from? This new gene, formed in the new chromosome, releases a protein called BCR-ABL in relation to the gene. This protein goes to the receptor called tyrosine kinase receptor, present in our white blood cells, leading to continuous activation of this receptor. This results in continuous proliferation of white blood cells in the bone marrow, causing a disease known as chronic myeloid leukemia. Chronic myeloid leukemia, or blood cancer. Then what can we do? How can we treat such disorder? The answer is very simple. This new protein, formed by this new gene, must be inhibited by any way. But how can we inhibit this protein? We will invent a new drug called, as you can see, Imatinibe. This Imatinibe inhibits this strange protein, preventing it from activating the receptor and stopping this continuous proliferation of these cells. Do we now understand the mechanism of action of this drug called Imatinibe? It's only one word. This Imatinibe and the family called tyrosine kinase inhibitors work by inhibiting these strange proteins. Not only this one. By the way, I only mentioned Imatinibe. However, we will design a new inhibitor drug for any strange protein that activates this receptor called tyrosine kinase. And then we will name this drug any name ending with NAB to join this drug family, tyrosine kinase inhibitors. This family inhibits this protein which activate tyrosine kinase receptor. What is the most well-known drug of them? Imatinib. What about its uses? It's more prominent in inhibiting this protein called BCR-ABL. And this protein doesn't exist except in patients having this Philadelphia chromosome that I told you about. Patients with chronic myeloid leukemia, even in acute leukemia sometimes, it's used in all types of leukemia. Now we have finished explaining this family and you now know this drug called Imatinib. But if someone wishes to disturb you, he would ask you about only one side effect. If you know it, then there is nothing more to be said. Tell him that it causes fluid retention and can lead to congestive heart failure. Leukemic patients on Imatinib are known to have swollen legs filled with fluid. We know that it's because this drug causes fluid retention, and these patients may become dyspneic and complain of heart problems. Now, let's shift to the next family. Family number 7 are called monoclonal antibodies. We only have 2 slides remaining, this and the next one. These monoclonal antibodies are considered the most recent item in the armory of anti-cancer drugs. And when you say monoclonal antibodies, you'll notice that the drug must end with MAB. So keep focused when we study any drug ending with MAB. Then this drug belongs to monoclonal antibodies family. But where did this idea behind them come from? Simply enough, scientists working on cancer research are studying these proteins present on the surface of cancer cells. Proteins present on the surface of cells are academically termed epitopes, like this. Epitopes. Epi means above. Epitopes are considered antigens, proteins that are present on the surface of the cells. Scientists are trying to research for any strange proteins that might be present on the surface of cancer cells. This strange protein would allow me to fish these cancer cells, because scientists can designate a special antibody for these proteins. Those antibodies are called monoclonal antibodies. Monoclonal means it targets only this specific protein. If I could hold a cancer cell and distinguish any strange protein on its surface and then design a drug which attaches to it, I would swear that this drug will only reach cancer cells, because this antibody is extremely specific. The best I can do is to look for a certain antigen or epitope present on the surface of cancer cells so I can target it. Among all different types of cancers, scientists have distinguished more than 30 or 40 different kinds of proteins present on the surface of cancer cells. For each type of these different proteins, scientists made a specific antibody that attaches to it. Now I'll show you some examples of monoclonal antibodies. Of course, the list is long, but as always, I'll tell you about the most common members. For example, we have this drug called trastuzumab. This trastuzumab attaches to a protein here on the surface of cancer cells. This protein is called HER2. That is its name. Proteins are termed with 3 or 4 letters. HER2 is short for human epidermal growth factor. Scientists discovered that cancer cells which release this protein called HER2 more than any other are breast cancer cells. Certain type of breast cancer called HER2 positive breast cancer. So when we have a patient on the oncology clinic, after removing her tumor with surgery, we send some samples of this tumor to the pathologist to determine whether these breast cancer cells are HER2 positive cells or not. If they are secreting this HER2 protein, then this cancer is called HER2 positive breast cancer. Needless to say, this patient soon after surgery should receive this drug called trastuzumab. This drug attaches to the HER2 protein. It can never attach to any other cell because there is no cell secreting this protein other than these breast cancer cells. On the same principle, we have a drug called rituximab that attaches to a specific protein called CD20, cell differentiation number 20. It's a certain type of proteins. Where is it present? It's present on the surface of B cells, immune cells type B. So all patients with B-cell lymphoma, the problem is in B cells. If you wish to target only this specific type of cells without any other cells from billions we have in our bodies, then you should use this drug called rituximab. This drug attaches to this protein called CD20, which is only present on the surface of B cells. You can target B-cell lymphoma, not only B-cell lymphoma, but also any disease related to B cells. Once you hear anything about B cells, remember this drug rituximab. Bevacizumab. On the same principle, it attaches to a protein called vascular endothelial growth factor. This vascular endothelial growth factor is present in abundance on cancer cells of colorectal cancer, colon cancer. Another drug called cetuximab attaches to a protein called epidermal growth factor. Same scenario occurs again. So instead of enumerating all these drugs present on the long list, that could be 40 or 50 drugs, remember that all of these drugs end with mab, and each of them is a monoclonal which is highly specific in its attachment to a certain protein present on certain cell. That's it. So all you have to do is to determine which type of cancer does this patient have, then you give him his specific monoclonal antibody. Social media these days are occupied with a new drug called dostarlimab. People are going crazy over this dostarlimab because it's assumed to achieve 100% cure of cancer. What is the story behind this drug? They say this drug was experimented on a group of colon cancer patients. They received this dostarlimab, which is a monoclonal antibody targeted to a specific protein called programmed cell death protein. When these patients received dostarlimab, nearly all of the 28 patients who received this drug were completely cured from cancer, and this made such a fuss throughout the world nowadays. Are we about to remove cancer from the world? Can we at some point design a highly specific monoclonal antibody that can attack only cancer cells without touching any other cells, and thus we get rid of cancer? Indeed, this is possible now. All we can do is to hope for this day to come. In general, this dostarlimab, which attaches to a protein called programmed cell death number 1 present on T cells, this drug is used for colorectal cancer. However, I won't dwell into details now, as this drug deserves a separate video specific for this drug. It has achieved 100% cure rate, so this drug deserves a special video with all possible time. But for now, let's be content with understanding the mechanism of these monoclonal antibodies. Now, all what could happen is that examiners who have been memorizing these drugs for 30 years or more may ask you simple question like this one. Which protein does trastuzumab attach to? The choices would be those 5 proteins. Now, you already know this trastuzumab attaches to HER2 and so on. That's why you need to memorize all these names and their proteins or antigens to which they attach. Their indications are not that important, so you only need to memorize these names and their antigens to which they attach. This is the last slide we have and we will be brief now. This topic is about miscellaneous or other drugs. We don't care about them all except for this one, drug called bortezomib. This is somehow new drug, as you can see it ends with mib. We have a very recent family ending with mib. So now we have a family ending with nib, which are tyrosine kinase inhibitors like Imatinibe. We have a family ending with mib. Those are monoclonal antibodies. And we have a new family ending with me. This is a new, yet unclassified family. It includes drugs such as bortezomib. Thus, they started the name with bortezomib. Bor because it contains boron element. And we don't see this element that much in pharmacology. We don't have many drugs containing this boron. And since this drug contains boron, then they named it after this element. So they named it bortezomib. This drug mechanism of action is not important. Not many people are interested in it. It works by inhibiting something called proteasome function. This proteasome function is one of the functions performed by cells, not only cancer cells, but also all normal cells. So it is just one ordinary biological function of cells. But for some reasons we don't know, we were surprised to see that certain types of cancers are highly sensitive to this drug, bortezomib. So when any patient receives this drug, all his cells are affected by it. But cancer cells in particular are very affected by this drug, especially multiple myeloma. So the most affected cells by this drug in the world are the cancerous cells of multiple myeloma. Unless examiners really hate you, they won't be interested in this drug. They know all students study the previous topics and no one of them cares about this one last page. This is the only reason for examiners to be interested in this part, thus you may see a question asking about the type of cancer treated by bortezomib. Pick this one called multiple myeloma. Another unclassified drug called interferon alpha. But if we wish to talk about this interferon, this is not the right place. It should be mentioned in another topic on another chapter because it is mainly used as antiviral drug. It's used against viruses. Indeed, it can be effective in some types of cancer, but it's not suitable to ask students in interferon alpha as an anti-cancer drug. I may ask you in it as drug treating HCV or other antiviral disease. So for now, don't bother with interferon. What we need from this page is only bortezomib that is used for multiple myeloma, and that's it. Now we have explained all anti-cancer drugs. And I don't think we neglected to mention any concept that an examiner at any place in the world might be interested in. It's not important where are you from, we covered all possible questions, all the possible ideas you may face in exams.